morning, everyone. My name is Mary Sanders, and this is for the class Intro to Cell Bio 2440 with Dr. Hudson. So my presentation is titled The Gateway for Proteins, and it's a look into the porrin protein. Alright, so before we can really dive into the porrin protein itself, I feel like we need to just really quickly address proteins, just to have a summary of that. So proteins are an essential part of life for all living organisms. They have tons of different responsibilities and functions, and some of those functions are digestion, movement, structure, support, communication on cellular level, and acting as chemical messengers. So now that we've basically just discussed proteins, I wanted to move on into one specific type of protein discovered by Hiroshi Nikaido. So Hiroshi Nikaido first discovered the porrin protein back during the E. coli outbreak in the 1970s, and because he discovered the porrin protein, they nicknamed him the porrinologist. So something really interesting about the porrin protein, in my opinion, is that it has channels that change depending on what type of molecule it is. So the channels can be bigger or smaller depending on the type of molecules it interacts with. So that was really cool. So the porrin protein was first found in the outer membrane of the gram-negative cell wall when Nikaido discovered it. But there are also now, we've seen that there's something called an aquaporin, and the aquaporin is found in the outer membrane of the gram-positive cell wall. So when the porins, though, are aquaporins, we know that positive membranes lack, or positive bacteria lack an outer membrane. So the porrin channels in aquaporin, they have to be bound to lipids in the cell wall as opposed to the porrin by itself. Then we know that porrin also is in eukaryotes like the mitochondria and chloroplasts. And the porins actually in mitochondria and chloroplasts are similar in structure and function to the porins in the gram-negative cell wall, and that is used frequently to validate the theory by Lynn Margulis, the endosymbiotic theory, because we know that in the endosymbiotic theory talks about that the mitochondria and chloroplasts developed and evolved from bacteria, and if in bacteria there are porrin, and then later on in mitochondria and chloroplasts there is porrin that's similar in structure and function, then we can logically conclude that, that jump made sense and that evolution made sense. So then we move on to figure 1 with our caption down below just for reference. And figure 1 is just describing the porrin protein through the ribbon model. So as you see here, we have the ribbon model and we have the different barrels, and they're color-coded just so that it's easier to see. And the porrin, well, I'm going to move on. Okay. So we know that porins are made up of beta sheets. They're formed by beta strands on the cytoplasmic side and loops of amino acids on the other side. We also know that amino acids on the outside, they interact with, so on the outside they're interacting with polar lipids, but in the inside there's polar residue, and it interacts so that the beta barrels are able to be aqueous in the middle, which makes sense because that's where like the transportation's happening. And there's a range of about 8 to 22 beta strands that make up each of the beta barrels in porins. I thought that's interesting. That's such a big range from 8 to 22. But yeah. Okay, moving on. So now moving on to the 4 protein structures. So we have another figure 2 in its caption, and I liked figure 2 because it kind of just lays out the structures in a very simple, easy to follow way, but we'll go over it. So we have 4 protein structures, right? We have, in all proteins, they have primary, secondary, and tertiary, and then if the protein has 2 or more polypeptide chains, then we have quaternary. So a primary protein structure is just a series of amino acids in a chain, and it kind of looks like a beaded necklace, in my opinion. Then we have a secondary structure that are these folded structures that form by polypeptides, and they're stabilized by hydrogen bonds. Secondary structure has the beta plated sheets, as we see here. It kind of looks like sideways Z's to me. And then we have alpha helixes, which look like ribbons or spirals. Those are the two different types that are in the secondary structure. And then we have tertiary structure, it's displayed here, and that's where one polypeptide chain called the backbone and it has like various secondary structures called protein domains. And tertiary structures are stabilized by hydrophobic interactions between nonpolar side chains, hydrogen bonding amongst polar groups, and electrostatic interactions between ionic groups with opposite charges. So tertiary is where polypeptide chains finally become functional. And then moving on to quaternary. Quaternary structure is basically the sum of all these like shapes put into one, and so it's like fine arrangement. And it's where the protein has more than one amino acid chain. And hydrogen bonding, salt bridges, and disulfide bonds are what stabilize the quaternary structure. And for my protein, the porn has all four of the structures. Moving on, we're now going to be discussing the two classes of porn. So porn proteins have two different functional classes, the general diffusion and the specific diffusion or specific porins. So general diffusion is where porins do not have specific preferences for the substrates, but sometimes they're like, it's not preferences, but it's just different permeabilities in cations or anions. But it's still not like specificities. But then with specific diffusion, it really does have specificities for substrates, and this is because the preferences are dependent on what size the porn proteins can hold. Because an easy way to think of it is it's kind of like this like little claw, and whatever space it has is what it can grab, and then it kind of transfers it like this, and then it releases it, and that's how it goes. It grabs and then goes off. So whatever it can hold is why it has specificities. But it also is what it can hold and what amino acids are involved with everything. Okay. Then we're moving on to porin's 5 superfamilies. So according to the Transporter Classification Database, or TCDP, porin has 5 superfamilies. So superfamily 1 comes from gram-negative bacteria. Superfamily 2 and 5 come from actinobacteria, and superfamilies 3 and 4 formulate from eukaryotic organelles. Now, porins have different pieces in them, right? Porins have a tilting angle, a shear number, a strand number, and a barrel radius, right? So we have all these pieces that make up this whole. So like tilting angle is the measurement of the angle made to the membrane. The shear number is how much of the amino acid residue is in each of the beta strands. The strand number is how many beta strands are in the porin, and the barrel radius obviously is just the radius of the porin's protein. So all of these pieces come together to make a whole, right? And we have these pieces making a formula. 2 formulas actually, and those 2 formulas displayed here, those are what help us be able to determine by plugging in our numbers and measurements and stuff. Those are what help us determine what type of porin we're dealing with, like what structure, what are the specific structures of the porin, because there are different structures, and so these 5 superfamilies and the different pieces help us be able to see that. So now we're moving on to figure 4. I really like figure 4. It is it lays out the DNA sequence arrangements of OMP-C of porins from Y, and Y is just talking about Yersinia, which is a genus of bacteria from the gram-negative species, and the OMP-C just stands for the outer membrane protein of C. And it goes on to lay out very nicely just like the DNA sequence. And as you see, there's things like numbers, colors. You see all these like L1, L2, and the L's are just talking about the loops. Excuse me, are just talking about the different loops of the channel, and then we have all these different colors like throughout here, and those colors are just representing polar, nonpolar, basic, and acidic residue. And it's just, this was so rare for me to find because even though there's been a lot of advancements in proteins and just like the information about that, for some reason there's very little that's been discovered about the sequence of homology of porins. So it was very hard for me to find like DNA sequencing stuff. So when I found this, I was like super excited, and I feel like it's just really interesting. And so I hope that more people will be interested in porin and want to dive deeper into it more than just about E. coli, because it's very fascinating, and we need more information like this and more coding and sequencing. But yeah, so in conclusion, porins act as the highway for all living organisms, right? So without porin proteins, there wouldn't be interactions between like the environment and bacteria, because like, there wouldn't be like bacterial cells diffusing through the bacterial membrane. Humans would be at a huge advantage without porins. I mean, porins are literally the channels that help us with diffusion, and it's important for things like antibiotics, which are needed to go through these channels to diffuse for the body. And that's huge for like medicine, the medical field, stuff like that. So porin is really important, and they just play a crucial role in making sure protein molecules are transported to where they need to go, and for allowing diffusion for bacteria and nutrients, so that bacteria can grow and evolve and stay healthy. So yeah, porins are super important. But that is the end of my slides, and then the following are just my literature cited, because I had a lot of references and figures. And that is it. Thank you.